The Part 5 plants notes will be on growth. Plant growth begins when germination of the seed occurs, which is shown in the image below. We have not talking about the process of germination yet, but we will come back to it in the Part 6 notes. But once germination of the seed happens underground, the plant will start to grow. And growth overall of a plant is considered to be indeterminate, which means for however long the lifespan of that particular plant is, its cells will continue to divide. And therefore, there's no predetermined growth that could essentially occur. It will just grow, 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 grow. Unlike humans, right, where you kind of grow until you are a certain age and then you don't really grow any taller after that point. But with plants, that's not true. They will grow for their entire lifespan. This does not mean they will live forever. Plants can live for a very long time. There are very old grasses, very old trees that are thousands of years old. But there are also plants that they only live for like 1 year and then they die, or only live for 2 years and then they die. And that's their normal lifespan. But however long their normal lifespan is, they can grow indeterminately within that time. Growth is confined to the meristems. The meristematic tissue is going to be tissue that is perpetually embryonic, which means it just continues to divide by cell division over and over and over again. These are the stem cells for plants. Meristematic cells are considered to be unspecialized or undifferentiated, so they have not yet become specialized to have specialized jobs. That will happen later when they are no longer meristematic cells, and they continue to divide near the points along the plant where the plant is considered to still be growing. So for example, an apical meristematic cell is going to be growing at the tips of the roots and the tips of the shoots. Axillary meristems are going to be stem cells that allow for lateral branching, so we could get growth right here. And there's going to be other types of meristematic tissue that we learn in this section of notes that will allow for the plant to grow wider overall within the stem, for example, like with trees. A plant is going to undergo 2 types of growth, primary growth and secondary growth. Primary growth is sort of the type of growth that we've kind of started talking about already. We are going to get elongation at the tips of the shoots and at the tips of the roots. This is going to be due to the apical meristems. So because of the apical meristems constantly dividing all the time, we are going to have plant stems that get taller, and then roots that get longer within the ground. Secondary growth is different. This is where we're going to get thickening or widening of both the roots and the shoots. So for example, trees are much wider at their base than they are at the top. So in secondary growth, the tree is getting wider and wider and wider. The stem gets wider and wider and wider. And this is going to be happening below ground as well in the root system. This is going to be due to a different type of meristematic tissue called the lateral meristems. Again, this will allow for the plant to get wider or thicker over time. That part of the plant will no longer be going through primary growth. Instead, it's going through secondary growth. And so that is kind of shown here, right? So here's the apical meristems. This is where, well, here's an apical meristem. This is an axillary meristem. But here's where primary growth would be occurring at the shoots, also here, and then at the roots. But secondary growth is going to be occurring near the wide part of the stem, and then also of the root system right here. And so that is going to involve a different type of meristematic tissue called the lateral meristems. We will talk more about the lateral meristems throughout the notes, but we have actually mentioned this one in a previous section of drawings, and I said I would come back to the vascular cambium. So it's going to become important in this section of notes. But this is also why, if plants are no longer going through primary growth here, right, they're growing through primary growth at the top and at the bottom, even laterally, but still only at the tips. They're not going through primary growth here anymore. These are mature cells that are now going through secondary growth. And so when that occurs, you're going to see thickening and widening of the plant here, but up here where the plant's still growing up higher, we're still getting primary growth there. So that's why, for example, on a tree, if you were to carve something, like if you carved your name at the bottom of the tree right here, well, if you come back, 10 years, 15 years, 25 years later, it is still going to be at that same position, right? It's not like all of a sudden your carved name is way up here. That's not how plant growth works, because down here is only going through secondary growth, so it's only getting wider or thicker, and the plant is only getting taller at the tips. Therefore, it would always remain in that same position. You should not carve your name in trees, though. It's harmful. Okay. Primary growth happens at the apical meristems, and this is going to be happening at both the root and the shoot tips. This picture over here shows in general how stem cells work. Remember that all cells, when they divide through a process called mitosis. Well, not all cells, there's also meiosis, but we're focusing on regular cells right now, and they will divide by a process called mitosis. When a stem cell divides through mitosis, it divides to make two cells, just like any other normal diploid cell would. It divides to make two cells, but when that division occurs, of the two daughter cells, one of them becomes a stem cell that replaces the initial stem cell that was now lost due to cell division, and the other one becomes a differentiated daughter cell over time. So this one gets to become mature and specialized, but this one stays a stem cell, and that way we constantly are refreshing or replying the plant with more stem cells. So a replacement stem cell is made, and then a daughter cell that can differentiate into specialized tissues. We will focus on primary growth in the root system rather than in the shoot system because it is more complex at the shoot than at the root. This is because at the shoot, there are going to be differences in gene regulation that occurs throughout a plant's life cycle that tell the primary growth areas that it's time to make leaves or it's time to make flowers. And so there's just a lot more complexities going on at the shoot tips. But at the root tips, we don't have that sort of specialization that needs to occur. So we don't have the same issues. So we'll focus on primary growth in the root system. When primary growth is occurring, growth occurs from the apical meristems, and ultimately this meristematic tissue will develop into additional meristematic tissue that eventually develops into specialized mature plant tissues. So we've talked about these mature plant tissues before: dermal tissue, vascular tissue, both xylem and phloem, and then ground tissue, the cortex and the pith. All of these initially came from meristematic tissue that ultimately went through a differentiation process to get them to be these more specialized mature tissues. Primary growth in the root system is concentrated near the tips of the roots. That's where the apical meristems will be. And this is going to cause the roots to move downwards into the soil. The tip of the root is going to be protected by a structure called a root cap. The root cap is important because it protects the meristem, the meristematic tissues, the apical meristems in the root tips from damage, because when the roots are being driven down through the soil, there's all kinds of things that could harm the root. And so we wouldn't want to harm the apical meristems down there, or it could lead to a halt in the growth or abnormal growth for the plant. So there's going to be like sticks, rocks, things like that that are very abrasive in the soil, and the root cap helps to protect it against that, kind of like a baseball cap protects your head from the sun. And then it also, in addition to protection, it will secrete a polysaccharide that will allow for lubrication of the root tip. And so as it's being driven into the soil, it will kind of help it to glide through the soil more smoothly. The root tip is going to contain 3 zones of cell that will be at successive stages of primary growth. So there's going to be 3 stages of primary growth. As a group of cells completes one stage of primary growth, essentially it gets moved up to the next stage. And so we're going to start with the zone of cell division, which is going to be closest to the tip. It's going to be at the root tip in the soil. So down here, protected by the root cap. So it is within these particular cells, the zone of cell division. It's within this particular zone that we do have our apical meristem. And so that's why there's lots and lots of cell division occurring here, lots of mitosis, rapidly. And so that's why this is called the zone of cell division. That's why we're constantly adding more cells to. Within this particular area, there is also going to be what's called a quiescent center. The quiescent center is a small group of apical meristematic cells that divide much more slowly. So this entire area is going to include the meristematic cells, but just this small little group of cells right here, that's the quiescent center. They divide more slowly than typical apical meristematic cells, and their job is to replace apical meristematic cells if the apical meristem gets damaged for some reason. And so here is a picture showing all the different zones. And so again, we have our root cap is down here, and then here's your apical meristematic cells, and the small little clump of these would be considered the quiescent center. If you are asked on an IB test to label the zones in order or to put the different zones in order, they will often include the root cap and the apical meristems within that vocabulary. And so the order is always going to be from the root tip upward towards the ground. It's always going to be the root cap, next the apical meristematic cells because they are close to the root cap. Then the zone of cell division would be labeled because it's a larger area. Then the zone of elongation, and then the zone of maturation closest to the soil. And in a stem, even though we don't talk about the details of the stem, these zones would be opposite, right? You'd have the zone of maturation closest to the soil. That's where all your mature cells are going to be. Above that, the zone of elongation, and above that, at the tips, is where your apical meristems are. And so that's where the zone of cell division will be, growing upward. So all the zones are opposite. Above ground in the stem. In the zone of elongation, that's going to be right above the zone of cell division. So once the cells are done dividing, they move up to the zone of cell division, and the apical meristematic cells continue to divide down here within the zone of cell division. In the zone of elongation, these cells are going to get longer. They can elongate up to 10 times their original length, and this is going to be due to a hormone that's called auxin. This is going to involve what's called ion uptake due to the presence of auxin. So ions are going to move in, we're going to have a loosening of our cell wall, water is going to enter the cells, and then the cell is going to get longer. It will elongate due to so much water being present inside the cell. I'll talk about this in greater detail on the next slide because this picture is going to be blown up bigger. But in addition to that, we'll continue to talk about how auxin works in regards to elongation all throughout this PowerPoint. Elongation is the primary driver of pushing the roots into the soil. It's not the zone of cell division that does that. The zone of cell division adds more cells, but it's the fact that these cells in the zone of elongation are getting longer that is pushing the root tip down into the soil. Okay, so what happens in the zone of elongation is the hormone auxin is going to stimulate a proton pump. And this proton pump is going to pump protons from low concentration to high concentration into the apoplast, or into the cell wall. Once these protons, these positively charged ions, have entered the cell wall becomes acidic, right? The presence of lots of hydrogen ions makes it a more acidic environment, decreases the pH, and that is going to activate a group of proteins called expansins. What expansins do is they weaken the crosslinks between the cellulose fibers. So you can see those crosslinks here that are like linking one cellulose to the next. It's going to weaken those crosslinks. And so once the cell wall now overall becomes weak due to loose cellulose fibers, this is going to cause water to be able to enter the cell. Once more water enters the cell elongates. Normally, the cell wall pushes back, right, and has all that pressure, that turgid pressure inside of it that wouldn't allow more water to go in. But by weakening the cell wall due to expansins, it does allow for more water to rush into the cell. And again, this will cause the cells within the zone of elongation to elongate up to 10 times their length, and that's going to push the zone of cell division further down into the soil. In the zone of maturation, so again, above the zone of elongation, once the cells are done elongating here, they move up to the region of, or zone of, maturation. And it's here where the cells are going to go through a specialization or differentiation process. So here's where they're going to go through changes in gene regulation that ultimately allow them to become specialized cells, right? So specialized xylem tissue, specialized root tissue, specialized dermal tissues. And so this is where these cells become mature and functional for the plant. So here's a picture again, just showing all those zones. So we have our root cap. Within this area, right above that's going to be your apical meristems. And your zone of cell division right above that. It's all kind of together. Then your zone of elongation where the cells get longer, driving the root tips further into the soil. And then here's our zone of maturation. These cells are much more mature, and they are able to have more specialized functions. Lateral roots are going to be those roots that branch off of the main root. So let's go back a slide, right? All these lateral roots, they are branching off of this main taproot right here. And so how do we ensure that they all stay connected to the vascular tissue? That's what the next slide is going to talk about. We must have a continuous flow of all vascular tissue into the stele. And so in order to make sure that happens, they sprout off of the pericycle. Remember, the pericycle is the first ring of vascular tissue, so it will connect to the stele. So you can see a lateral root is branching off right here, continue to get longer down here by primary growth, but we will be able to have that connection into the stele because it comes off of the pericycle. Secondary growth is different because it results in thickening or widening of stems and roots, and it's going to occur in nearly all, well, in all phylum of plants that have trees specifically. So for example, gymnosperms, these are your conifers belonging to phylum Coniferophyta. So those are going to be like your cone bearing plants. Like a redwood tree, for example, some type of pine tree. And then it's also going to occur in many dicot angiosperms. Monocots are not going to go through secondary growth. Only dicots are going to go through secondary growth. So think trees, for example, which should make sense because an example of a dicot is like grasses, right? And they don't get any wider or thicker. So focus on tree type of plants. And so this is going to be the result of the lateral meristems. And there are two lateral meristems. our cork cambium. The cork cambium is going to be responsible for adding more dermal tissue. Eventually, this will form bark. And then the vascular cambium is responsible for adding more vascular tissue. So it's going to lay down new xylem and new phloem, but mostly xylem. It's definitely going to lay down a lot more xylem than phloem, which ultimately will become wood. So here is our vascular cambium. We actually drew this in an early diagram in the part 2 notes. And so you can see here is our xylem. It's going to be laid down towards the inside, and then our phloem towards the outside. So it is going to make both secondary xylem and secondary phloem, but not at an equal ratio. It will make more secondary xylem compared to secondary phloem. So as the layers of secondary xylem accumulate, which they will accumulate over time, ultimately they will become wood. And so this is going to consist of mostly tracheids and vessel elements and fibers that are able to kind of harden over time and give wood a really strong, tough structure. The cell walls also have a lot of lignin in it. So again, leading to strength and hardness for wood. Wood is such a strong, durable material. That's why we use it to build with. And it's this lignin and these other components to it, these other tough structural components to it that lead to that. But we can see the annual growth rings in wood. This is all like an accumulation of secondary xylem. The primary xylem is like way in the middle here. There's not very much of it anymore. It's really mostly secondary xylem. All this is secondary xylem. So then again, secondary phloem, where would that be? Well, that's going to be not shown in this picture. It's going to be like way on the outside. So in this picture, it's shown in pink because here's our vascular cambium in black. And so this is going to be our secondary phloem, and it doesn't accumulate extensively over time. Ultimately, it becomes part of the bark and eventually just kind of like sloughs off the tree and gets like slowly replaced by the vascular cambium, whereas the secondary xylem gets a lot more of it made. And so that's why you see like so much of the tree being that hard wood near in all the stem, right? It's that really hard wood, and it accumulates a lot more over time compared to the phloem. The next part of the notes will get into more details about the hormones involved in plants and how that leads to tropisms. Plant growth, development, and responses to stimuli are controlled by plant hormones, and plant hormones are called phytohormones. Okay, phytohormones are the hormones found in plants. These are the particular phytohormones that we're going to cover in this class: gibberellins, auxins, cytokinins, ethylene, and abscisic acid. This chart just kind of gives a general idea of what each of these hormones does. You don't need to memorize it. I'll go over the details, but it does kind of give you an idea of where we're going. What a hormone is it's a chemical messenger. Within the human body, we have hormones too, and that's one way for us to send a message to trigger a response inside the cell, and that is always going to be a chemical message. We also have the ability to send messages through neurons, right? So we can send electrical messages also. But plants, they don't have a nervous system, so all of their responses are sent chemically by hormones. And so the hormone is typically produced in one part of the plant, and then it gets released, and it can have an effect on the organism in a completely different part of the organism. So we can send it through the phloem tissue to other parts of the body, or plant, sorry, and ultimately it can trigger a response there. It typically involves what's called signal transduction, which we did talk about a little bit in Bio-H, and you will talk about it a lot more in IB Year 2. Signal transduction pathways involve something like a hormone right here bonding to a receptor in the membrane, and notice it has the right shape. It's kind of like a lock-in key. So this receptor protein in the membrane is ready to receive this chemical message from this hormone. The hormone itself does not come inside of the cell. It doesn't need to. Instead, it sends a chemical message, and that's going to go through a process called signal transduction. It is kind of like a domino effect where one protein sends a signal to another protein, sends a signal to another protein, until ultimately it's going to tell the cell to do something. It's going to activate some sort of cellular response inside the cell. Again, that's called signal transduction. We will talk about these hormones up and coming in this set of notes. The main hormone we're going to focus on, there's a lot of details to focus on this particular plant hormone, and it's called auxin. There are lots of different kinds of auxins in the plant cells. That means auxins do have a lot of functions for a plant. One thing they do is they initiate growth at the roots and at the shoots. So we talked about that, right? We said that auxins are going to be used to be able to maintain what's called apical dominance. We're going to talk about that more. It also is used, which we talked about a little bit, in cell elongation in the roots and the shoots. It also does influence the development of fruit, not the ripening, but the development. So that means like the fruit forming, maturing, getting bigger over time. And then the same for leaf development. So it's responsible kind of for all of those things. IAA is the most common type of auxin, and it is the one that promotes the elongation of cells in stems. So we've already looked at this type of auxin, and we'll continue to look at how this type of auxin works in more details. It's made in the apical meristems of the shoots, and it gets transported down the stem to stimulate growth of the overall stem. That was going to be used in elongation of cells. We'll come back to that more. And then at very high concentrations, though, it will actually inhibit growth, and this helps it to maintain apical dominance. So let's take a look at this in more detail. This picture again shows you how auxins are used to stimulate cell elongation and therefore lead to growth. Auxins are going to stimulate the proton pump to become active. We're going to pump hydrogen ions from low concentration inside the cell to high concentration in the cell wall. And then that is going to decrease the pH, make it more acidic, which activates expansion proteins. Expansion proteins are going to weaken the cross links between the cellulose fibers, which is going to allow for water to rush into the cell. Once the cell is filled with water, now the cell can elongate because it has those weak cell walls. So that leads to growth of the stem. So in this picture right here, this shows the apical meristem, and it's growing up, but imagine that this apical meristem was still down here, right? It used to be down here, and now it's moved up in this direction. Well, when it was down here and it started to grow up, it actually like leaves a little chunk behind. So these little chunk behind, they were formed from the apical meristem, but now they're like tucked in to this, little location at the nodes, between like right at the nodes. And we've talked about this little meristem before. We called it the axillary meristem or axillary buds. These axillary buds are made from the apical meristems. They contain incredibly high concentrations of auxins because of that. And so that is inhibiting their growth because there's so much auxin here. In addition to that, auxins from the apical meristems are sending down more auxins, telling these axillary meristems don't grow. However, the higher up the apical meristem moves, like as it's growing upward in the stem, the farther away it gets from these axillary meristems, and so the amount of auxins that make it all the way down from the shoot tip to the axillary buds is less in concentration. And so axillary buds that are farther down the plant will have some release of apical dominance. And so that's why you see them they start to grow, right? So For example, you might see more of these branches coming off the stem down here at the axillary buds because it's farther away from the auxins at the shoot, so less and less of it is actually making it down. And so there's going to be some release of this apical dominance down here. And so because of that, you get lateral growth. So lateral growth in regards to branching. So the plant's going to get a little bushier towards the bottom. In addition to release of apical dominance as the shoot tips grow farther and farther up, you also have this other hormone called cytokinins that are made in the roots, and they actually are sending the cytokinins up. So they actually help in root growth, but in addition to that, they also are going to move upward, and they are going to kind of counteract the auxins. So the auxins are coming down from the top saying, don't grow to the axillary buds, but the cytokinins are coming up saying you will grow to the axillary buds. And so it's this ratio between cytokinins and auxins that are going to determine whether the axillary buds will grow into lateral branches or not, right? And how much growth they will occur. There's a balance of this ratio. So again, the plant tends to be bushier towards the bottom than at the top. Gibberellins are a plant hormone that are also produced in the apical meristems, and they actually have many roles in a plant, but IB is going to focus mostly on their role in stem elongation. So they are going to help in cell division and cell elongation, but they're going to do it differently compared to auxins. You don't need to know how. So unlike auxins where you have to know that mechanism, you don't need to know the mechanism for gibberellins. You just need to know that they do help in cell division and cell elongation, and that it is different in how they operate compared to auxin. In this particular graph down here, you can see that we're increasing the concentrations of gibberellins given to the plants, and so based on that, we're seeing more growth occur. More primary growth occurring. And so you should be thinking about, is there anything in plant hormones that I could be interested in doing an IA on? Because we can get some of these hormones. Like we have gibberellic acid, which is gibberellins here at Newbury Park High School. So potentially, could you do an IA using plant hormones? Some food for thought. The next hormone we'll talk about is ethylene. Ethylene is a gas. It's a volatile gas, which means it easily vaporizes. And it does have a different name in chemistry. Chemists are going to call it ethene. Biologists are typically going to call it ethylene. It does say in the IB standard that you need to know both terms. So be prepared to recognize both these terms, ethylene and ethene. Ethylene gas stimulates fruit ripening. And this is an example of positive feedback, which means that whatever the initial stimulus is, ultimately leads to more of that stimulus being made, right? Intensifies that stimulus. It's positive feedback. And so in this case, ethylene produces ripening, leads to ripening in fruits, but as a fruit ripens, it makes more ethylene. So again, positive feedback. Ethylene can actually diffuse into other fruits of the same species or of different species and initiate their ripening. And once ripening becomes initiated, more ethylene will be produced since it is a positive feedback loop. And so what this leads to is synchronized ripening, where all the fruits kind of come ripe all about the same time. And this is an advantage to both farmers in agriculture and in nature. So farmers are going to force the synchronized ripening of their fruits because they want to be able to unload all the fruits from their trees, like harvest all the fruit, and then, ship it to the different grocery stores and have it ripen all at the same time so that they can sell their product, right? When it's nice and ripe. Versus if they had to, pick at different times, it would kind of slow down their overall ability to be profitable. right? So they want to be able to maximize what makes the most sense. And so having the fruit get ripened all at the same time is a good advantage. They can also, when they ship the fruit, sometimes they have to ship the fruit like really far locations, like across the country, or even like in huge ships across the ocean to other countries. And so in order to last that transport, they might want their fruit to not ripen very much at that time so that it can ripen at the grocery store. So within these trucks or ships, they can actually like kind of vacuum out the ethylene gas as it's being produced by the fruits, and this will slow down that ripening process so that it can make it all the way to shipment without spoiling. They can also do the opposite if they wanted it to continue to ripen along the way. Like if it's a slower ripening process, they can like pump ethylene gas into it, and that would like force the ripening faster. It's also used in nature because as one fruit becomes ripe, it leads to the next fruit becoming ripe, and so on. And so again, synchronized ripening would happen in nature. And this is important because then it attracts all the animals. So then if we have all this nice ripe fruit, all the animals come, they eat the fruit, and then they carry the seeds away and typically eliminate the seeds in their waste. And so it plays a really important role in seed dispersal. These two tables up here shows you that some plants are ethylene producers, which typically mean that they produce the ethylene and they're sensitive to ethylene, right? So they make ethylene themselves, and then that leads to more ethylene being made, which allows for that fruit to ripen in a synchronized way. But there are other plants that don't necessarily produce ethylene. However, they are sensitive to ethylene. So if you put a plant that is an ethylene producer near a plant that is ethylene sensitive, even though this particular species of plant does not make ethylene themselves, it will cause these particular fruits to ripen. So that's why you often see like maybe if you put your bananas, for example, bananas are ethylene producers. If you put your bananas near your grapes, it would cause the grapes to ripen faster. But grapes themselves don't produce ethylene, so they don't ripen off the vine without this. Or that's also why sometimes you see people putting their fruit that are ethylene producers in bags to make them ripen faster. For example, avocados are very hard. You can't eat them unless they're perfectly ripe. And so people will buy them at the grocery store hard and then put them in a bag. And that kind of helps to trap some of that ethylene in there and help their ripening. So just kind of making it relatable. The last hormone we're going to talk about is abscisic acid, or ABA. Abscisic acid is going to be involved in two areas of the plant: seed dormancy, which we're going to come back to later in the Part 6 notes, and then also in response to environmental stress. ABA is produced in the roots, and it's kept in the roots unless it is needed as an emergency response in the guard cells. So if the roots sense that it's incredibly dry in the soil, and therefore the plant's not getting enough water, then ABA translocates from the roots to the leaves, where it rapidly alters the osmotic potential of the guard cells. And once the osmotic potential of the guard cells is altered, it's going to cause the water to leave the guard cells. Then the guard cells become flaccid and it closes the stoma. And this happens very fast because it's an emergency response. This is not just like how the stoma are regulated on a regular basis. We already talked about that, and that involves just ions that are always around. This is different because it's a rapid response, because it's like the emergency shutoff. It's very dry, not getting enough water. Plant will die if we don't close the stoma. Send the ABA up to close the stoma. Tropisms are when plants respond to their environment. So we're going to talk about different types of tropisms, specifically in the stems and in the roots, like why each one of them grows up versus down. There are positive and negative tropisms. Positive tropisms means that growth occurs towards the stimulus or with the stimulus, whereas negative tropisms means growth occurs away from the stimulus or in the opposite direction of the stimulus. There's different types of tropisms, and we are going to focus on only two. We are going to focus on geotropism, which is or also called gravitropism on an upcoming slide, which is a plant's response to gravity. And then phototropism, which is a plant's response to light as the stimulus. But here's some other tropisms too. Hydrotropism, that's how it grows in response to water, because plants need access to water. And thigmotropism is kind of neat because this is how like vines are able to like grow up and wrap around things, because thigmotropism is response to touch. So as it touches whatever it's trying to climb up, it will cause it to like wrap around that substance, like a trellis, for example. Phototropisms are when plants grow towards light. And so stems are always going to grow towards the light. And this is because they are positively phototropic. So we're going to talk about how that happens in more detail. But again, they're going to grow towards the light, with the light, towards the stimulus, which is what makes them positively phototropic. If you were to put a plant in the dark, though, for a while, the stem itself is still going to grow up, even though it doesn't have access to light. But this is going to be due to the fact that the stem is negatively gravitropic. So it's going to always grow opposite of gravity. Gravitropism or geotropism is growth in response to gravitational force. So we already said that that the stems are going to be negatively gravitropic because they grow up when gravity is pulling it down, but the roots are going to be positively gravitropic because as gravity is pulling a force that pulls down, and remember the roots are going to grow down with gravity. So they are positively gravitropic. We're only going to learn the details of phototropism. We're not doing details of gravitropism. Auxin plays a major role in phototropism. And this is like a pretty complicated process right here. We already know that auxin is going to be used in elongation of developing shoots, right? So we've talked about that process already. But this is going to be how light is going to also be involved in that particular response. So phototropins are proteins that are going to be in the cell membrane or plasma membrane of the stem cells, and they are going to be stimulated when a certain wavelength of light hits it, blue light. When blue light hits it, it's going to go through a conformational change, so a shape change. Remember, conformation means shape. So without light, it would be shaped like this. With light, shape change. You don't need to know anything about this shape change. You just need to know that it does undergo a shape change when blue light hits it. In the dark, it won't go through any conformational change. Therefore, we're not going to get any change in gene expression of any of the proteins involved in this process. But when blue light hits it, we will go through a conformational shape change. The phototropin is stimulated by light. That goes through a shape change, and that is going to activate a protein called the PIN3 protein. So we're going to get a change in gene expression of the PIN3 protein. What the PIN3 protein does is move auxin out of the cell. So the PIN3 protein is considered an auxin efflux carrier protein. So efflux means it's leaving, right? It's exiting. Auxin is exiting the cell through this particular protein. It is an efflux protein, transporting auxin out of the cell. So what we're going to see is the sun is going to activate that phototropin, which is going to activate the PIN3 protein, and that is going to cause the plant to sort of bend towards the light. And this is because we're going to be moving auxin out of the cells that are on the lighted side. So we're going to look at that. One thing about auxin is that it can diffuse passively into cells. So remember, passive transport means it's moving from high to low concentration and no energy is going to be needed. And so the blue proteins right here are going to show that. These are going to be channels that are called auxin influx channels. Oh, sorry, they're the orange ones, not the purple ones, the orange ones. The orange ones are going to be our auxin influx channels. They're going to move auxin from high concentration to low concentration, so not use any energy. But they're only going to do this when auxin is undissociated. So auxin has to be in this form. It has to have its full carboxyl group. It cannot be in this acidic form. So as long as it's in this form, it gets transported into the cell, no problem. Once it's in the cell, though, it's going to lose a proton. So once in the cell, auxin loses the proton, it becomes dissociated. That's what that means. And so it now has this charge. Once auxin becomes charged and acidic inside the cytoplasm, it gets trapped. Okay, so the negatively charged auxin gets trapped inside the cell, and in order to get it to the next cell, we have to pump it. We have to pump it out using our PIN3 protein, our auxin efflux carrier. So those are going to be shown in purple. So that's our PIN3 protein in purple. Its job is going to be to move that auxin back out of the cell. Auxin is exiting the cell, efflux of auxin. Then auxin is going to make it into the apoplast, and the apoplast is acidic. You can see that pH here. And so once in the apoplast, it picks up a hydrogen ion again, and then it becomes this form again, right? Full carboxyl group. It's no longer negatively charged, which remember means it can go back into the cell without using any energy, using the influx protein. And it just keeps doing this, okay, over and over and over again. So in the cell wall's acidic, picks up a proton, not acidic anymore, gets influxed into the cell without using any energy, loses a proton in the cytoplasm, becomes acidic, gets trapped, has to move by efflux proteins, our PIN3 protein, using energy against its concentration gradient, back into the cell wall, and then repeats the cycle. Influx, efflux, influx, efflux, over and over and over again. That's how we transport auxin from one cell to the next. So let's see why we're doing this. If all the auxin efflux carriers, so all the PIN3 proteins, concentrate on one side of the cell specifically, so if we go back a slide, right? So you can see like they're all on this bottom side of the cell, bottom side of the cell. So if they keep doing that, if they keep going to one side of the cell specifically, we can keep transporting auxin from cell to cell to cell and maintaining an auxin concentration gradient. This is important because, remember, we have light coming in, stimulating our phototropins. They go through a shape change, and that is what's going to lead to auxin being transported from cell to cell to cell. This is going to be a lateral transport. Auxin is going to be transported from the sunny side of the stem to the shady side of the stem. So if you go back a second and we look at this, right, we have our auxins. We're looking at this as if it's going like from up to down, but if you imagine instead, if you like rotated this like horizontally, so that it was like cell, 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 right? And this was our sunny side, so here's our sunny side. The sunny side has caused all this activation, and what we're doing is we're transporting auxin from cell to cell to cell to cell from the sunny side of the plant to the shady side of the plant, because all our efflux carriers have concentrated to do that for us. So we get this concentration gradient that moves the auxin to the shaded side of the plant. Then, once auxin gets to the shady side of the plant over here, that's when it goes through this. Auxin stimulates the proton pump. We pump hydrogen ions from low concentration to high concentration, activates expansins, weakens the cell walls, water enter, the cell elongates. So notice the cells are elongating on the shady side of the stem. This is because we've moved all the auxin here from the sunny side. Therefore, we're getting no growth on the sunny side, and lots of growth, elongation, on the shady side. Therefore, we get a bend. It's going to bend towards the light, right? So this is positive phototropism. positive phototropic response in the shoots because of auxins. Auxins are stimulated to move laterally from the sunny side to the shady side, where they are able to promote cell elongation on the shaded side, causing it to bend towards the light, right? So now it's going to have more access to lights because of this. So that's a pretty big advantage of it. If we were getting equal concentrations of or equal amounts of sunlight coming down from, like, let's say the sun was directly above the plant, then you're not going to get this bending, because instead of having the auxins move to one side, like it's shown here, all these little dots are auxins. Instead of getting the auxins move to one side, the auxins are going to be distributed equally down both sides of the plant, and that would cause the plant to grow straight up. Okay, so you only get the bend if auxins are heavily transported to one side of the stem, because the light is going to be on just one side rather than right above the plant. And again, this is showing that. So here's our sunny side of the plant, right? The sun activated our phototropin proteins that caused a conformational shape change, which activated or change in gene expression of our PIN3 proteins. If all the PIN3 proteins move to one side, right? So this side, this, if we had multiple layers of cells here, like in this picture, right? If they all move to like this side, this side, this side, this side, this side, then we keep moving the auxins along, right? Influx, efflux, influx, efflux, influx, efflux, all the way across until ultimately we get it to the shaded side of the plant. Once auxins are in high concentrations, they will stimulate the proton pumps. Protons will move into the cell wall, activating expansins, weakening the cell wall. Water rushes in, elongates the cell, bends towards the light. So we're only going to get this bending because the light is unevenly distributed. I'm going to go back a slide really quick to show you this picture. Like if the sun was up here, right above the plant, then we wouldn't get this lateral transport of auxin over here. Instead, auxins would go on both sides of the stem equally, and so that would just cause the stem to grow straight up. Okay, so positively phototropic. And that's it for the part 6 or 5 notes.